after the class, I'll make sure to post this up on the Google Classroom so that anybody that's interested can go back in and rewatch it, catch up on any parts that you missed, so on and so forth. So let's go ahead and jump into the classroom here. This one? Yeah, this one. So just as a reminder, something that Naya brought up earlier, the assignment did get posted for today. So this is that last assignment that we're doing at the Excel Center to try and get writing submission from students to kind of look at people's writing over time while they're here at the school. So this is the one that is about I'm writing about past experience with science classes, either here or other places. And if you haven't taken a science class in a while, then some stuff about what you're interested in this class in biology. So again, we do want people's authentic writing samples. So it should be on a handwritten piece of paper. So just like a blank piece of notebook paper like this one. Please make sure to have your name, your full name, so first and last name, and date at the top of the paper before you send it to me. Otherwise, I'll have to ask you to resend it in with that information on it because they want that info on it when we submit it to the Excel Center. And then you can turn in your writing sample by either uploading it on the Google Classroom. You can take a picture of it and text it to me or email it to me, whatever works best for you. Again, nobody's going to be like critiquing you and sending you comments about your writing. We're not looking at it to criticize people on their spelling or grammar. We just want it in sort of a more holistic sense to kind of gauge people's writing abilities as they go through their classes here at school. So as long as you get it turned in to me, then you'll get full credit for having it turned in. I'm not going to take points off of anybody's for it being like too short or you misspelled a whole bunch of words, your grammar wasn't very good. Don't really care about that so much as that you just write it down, put your name and date on it, and send it in to me.
Does it have to be a full page?
Or does it have to be like in a white paper?
If you can make it close to a full page, that would be preferable. Mostly we just don't want people sending in like 2 or 3 sentences. So if you can make it to a full page, that would be great. But somewhere between half a page and a full page is kind of what we're looking for. We basically want there to be enough writing so we can kind of get a good sense of where your skill set. And yes, in response to Kevin's question, who's here with me in the class, it does have to be on like a piece of paper written down because we don't want you guys getting extra help from like spell check and autocomplete and stuff like that. And so in your own handwriting on just a regular piece of notebook paper or printer paper, something like that. And then again, once you're done with it, just take a picture, send it to me, and you'll be all set. All righty. So with that taken care of, let's jump into the notes for today. So like I was saying earlier, today we're going to be talking about what's called the scientific method. Basically, it is a series of steps that scientists use in order to come up with ideas for experiments to do to test out the questions that they have about the world around them. So there's this kind of general series of step by step that scientists go through when they're trying to figure out a good way to answer a question they have about the natural world. So we're going to go through kind of each step individually and talk about what it means and how it works. But just to kind of give you guys an overview of everything that we're going to be going through today. So the scientific method starts off with some sort of observation that we make about the world around us. That leads to a question, something that we were kind of wondering about, thinking about. And based on that question, we can form what's called a hypothesis, which is like an educated guess or prediction about what we think the answer to the question is. In order to see if our hypothesis is right, we have to do an experiment, a well-designed experiment that'll give us good data, good observations that we can use to answer our question. So as the experiment's going along, we're making observations, we're collecting data, we're writing down what we're seeing, and then we can use this data that we collect to come to a conclusion. Something that tells us about what is the answer to our question, and was our hypothesis correct? Did we correctly guess what we thought was going on in nature? And then finally, if you want to get really, really good data, you're going to do the same experiment multiple times. That way, you can say, I did this experiment 50 times, and I got basically the same result every time. That makes your experiment really, really scientifically strong. So, starting off first with making observations. So when most people, you think about observing something, most people will think about like seeing something, right? Like I saw this car or I saw this person walking by, I observed them walking. So that's definitely one way that we can make an observation. But any of your senses, any of your 5 senses can be used to make observations. So you can also hear things, right? So you can hear if something is this direction or that direction. You can hear if it is far away or if it's close. You can hear if it's loud or quiet. Those are all observations you can make with your sense of hearing. You can touch things. Like I can feel that this desk is really smooth and hard. Same thing with my computer right here. But the paper over here is smooth, but it's a lot more flexible and bendable. Those are things that I can observe with my sense of touch. There's also your sense of smell. So you can use this to observe, is this milk good or has it been sitting in my fridge for a couple weeks and it's gone bad? Can I smell something really sweet or sour or bitter, kind of off in the distance? And finally, there's your sense of taste, which hopefully you're mostly using to observe things about food and not anything else. But you can taste if things are sweet or salty, if they're really savory or really spicy. Those are all observations that we can make about our natural world. So again, science is all about studying the world and the universe around us, whether that's on a huge planet, galaxy scale, or down on a tiny microscopic scale, or anywhere in between. That's what scientists are interested in. And so we use, honestly, mainly in science, we use a lot of visual observations, a lot of measurements and stuff like that. But we can also use those other senses to gather observations as well. So one example of an observation that we'll kind of use to base our scientific method discussion around for today is that imagine that you're going on like a walk through the forest or through like a greenbelt area in Austin. And you notice that there's some ponds that are around, and these ponds have salamanders in them. If you've never seen a salamander before, it's like a small lizard that likes to live close to the water. So they're related to things like lizards and frogs and stuff like that. So much like lizards, salamanders, for the most part, are supposed to have these like nice, long, straight tails. Some of them are really thin and wispy. Some of them are kind of short and chubby. But for the most part, they should be straight in one kind of line coming off the butt of the salamander. However, you notice that some of the ponds have salamanders that have these weird kind of wonky, curvy tails. They're not supposed to be like that. They're supposed to be nice and straight. But something has happened here, and you observe these salamanders that have these weird curved tails to them. So you're thinking, that's not right. They're not supposed to look like that. Something must be happening here that's causing these salamanders to have these weird curvy tails. So you observe the weird tails, and that could lead you to a hypothesis. So your hypothesis is like your suggested solution or your suggested answer to your observation or to your question or to your problem. So you don't know what the answer is yet. We don't know why these salamanders have weird curvy tails. But the hypothesis is us saying maybe this could be the case. One important thing to remember about a hypothesis, or in the plural form, if you're making a lot of hypotheses, is that it must be something that is testable. That means something that we can do an experiment on in order to test. So some things that are not testable would be things like, I think these salamanders have curvy tails because the gods of the sea have created this new species. That is a fine hypothesis, but you can't really test if the sea gods created these new salamanders. Or say, oh, these salamanders obviously have curvy tails because Mars is in ascension right now, and the Sirius star is on its fourth plane, and Venus is aspected near the sun at this time of year. So all that together means that the salamanders have weird tails. Again, that's all great. You can't really do a test on things like astrology and horoscopes and stuff kind of like that. So in order for it to be a good scientific hypothesis, it has to be something that we as humans can do experiments on and gather data about. One way to kind of think about hypothesis statements that I find helpful, especially when you're just kind of starting off thinking about them, is that sometimes they're written as if-then statements. So if I do this thing, then I will see this observation again or this result again. So for example, if you're interested in seeing if a fertilizer will affect the growth of a specific kind of plant, then your hypothesis could be something like, if I add fertilizer to this cactus, then it will grow bigger than other cacti. So if I do this change, if I do some experiment, then this is what I think will happen. These are the results that I think I'm going to get. So a hypothesis is sort of like a guess, and it's sort of like a prediction. You're predicting what you think the answer to your question is, what you think if you do an experiment, what the answer could be. So if we think back to our salamander example, we might say that we think that the salamanders have curved tails because there's some sort of pollution in their pond. There's some sort of pollution in the soil where they live. You can also phrase it as, if salamanders grow up in a polluted environment, then they will grow weird curvy tails. So that is our hypothesis. That is our guess or our prediction about what do we think is causing these weird curvy salamander tails. So in science, a hypothesis is like a scientific guess or a scientific prediction. It's what we think is going on, and it's something that we can test using an experiment. All right. Does anybody have any questions so far about observations, hypotheses, coming up with a testable hypothesis? Any questions on anything so far?
I'm good, Mr. Mike.
All right. Cool. Thanks, Nasima. All righty. So we have our observation, our thing that we saw or smelled or touched. We have our hypothesis, what we think is causing it. But if we want to get some answers to our hypothesis and figure out if our hypothesis is right or wrong, we need to do an experiment. So an experiment is a set of procedures to test the hypothesis. We want answers, dang it. And we are going to do something in order to figure out, was our hypothesis correct, or were we off with our hypothesis? Was it wrong in some way? In order to get that information, we need to do some experiments. So each experiment that scientists do is made up of a set of things called variables. So variables is essentially just kind of a fancy scientific way of saying things that can change in an experiment. Different things that could affect the outcome of your experiment are called variables. So there are things that we are trying to change as the scientist. There are things that we are kind of measuring or observing as part of the experiment. And there are variables that are kind of happening around us that we're trying to keep control of or keep track of. So the first rule of doing an experiment, of planning a good experiment or a valid scientific experiment, is that you can only have one changing variable at a time, or at least you should try your best to only have one changing variable at a time. And in order to kind of show why this is, let me set up an example for you guys. So imagine that you're doing the salamander experiment. You think that pollution is causing the salamanders to have the weird curvy tails. So you set up two different chambers. And the first chamber, you set it at 77 degrees, nice and warm. You set it at 80% humidity and make sure that there's plenty of water kind of on the ground and in the experimental chamber. And you make sure that it is free of pollution, no pollution in the first chamber. Put some salamanders in there. And then in our second chamber, we're going to keep it a little bit cooler, down at like 60 degrees, so a little bit on the cool side. We're making it super dry. We're taking away a whole bunch of the humidity and limiting the amount of water that's going in there. And we're adding a whole bunch of pollution to the soil. So we have a warm, humid, non-pollution chamber and a colder, dry, pollution-filled chamber. You do the experiment, you let the salamanders grow up, and you see that in chamber number 2, you have a whole bunch of salamanders that have a weird, crinkly, curled tails. So based on your experiment, can you say for sure that pollution is what caused the salamanders to have the weird tails? Feel free to answer either out loud or you want to put your answer in the chat. That works too. But based on that experiment with the two different chambers, can you say 100% for sure that it was the pollution that caused the salamanders to have the weird tails?
Mr. Michael, can you please repeat it, like the explanation, please?
Sure thing. So you have two like experimental chambers, experiment areas. One of them is warm. It's got lots of water and lots of humidity, and there's no pollution. And the second chamber, it's colder. There's less water, very dry, and there's pollution in the soil. You put a group of salamanders in each one, let them grow up, and the second chamber, the colder, drier pollution chamber, has crooked-tailed salamanders. So based on those two chambers, hot, humid, wet, no pollution, cold, dry, less humidity, more pollution, do you know that it was definitely the pollution that caused the change in the salamanders? Anybody have any thoughts they want to share? We said that it was maybe the pollution. I mean, that's our hypothesis. That's our guess. But was the pollution the only thing that was different about the chambers? Mhm. Yeah, exactly. So as Kevin just pointed out, the pollution is not the only thing that's different about the chambers, right? They had different temperatures on them. So the salamanders in the one chamber were a lot colder than the other ones. They had different humidities and different environments. So the salamanders in the first chamber had a lot more water and humidity than the second one. So because these chambers were so different, we don't know for sure what caused the second chamber to have the curved-tailed salamanders, right? Sure, maybe it was pollution. That was something that was different between them. Wait, but also maybe something about the temperature affects the way that tails grow in salamanders. Or wait, maybe having less humidity causes something to happen in the salamander body and it causes their tails to grow differently. So because they're in such different environments, we don't know for sure what it was that caused the salamanders to grow with the weird tails. So now imagine that we go back and do the experiment again, but differently. This time we set up both chambers at the exact same 77-degree temperature. Both chambers have a high humidity and plenty of water, but only the second chamber has polluted soil in it. The first chamber has no pollution. So we do our experiment again, and we see that the second chamber now has the crooked-tailed salamanders, the curvy-tailed salamanders. Now, we have a much stronger case that it's the pollution that's causing this issue, right? Because the temperature was the same in both places. The humidity was the same in both places. The access to water was the same in both places. The only thing that we changed was the pollution. And sure enough, when salamanders grew up in the polluted chamber, they ended up having the crooked tails. So it makes it a lot easier for us to say we kept as much things the same as possible. And when we changed the amount of pollution, we got the salamanders with the weird tails. So by only having the one variable that we're changing, by only changing that one thing in our experiment, it helps it make it much more clear what is causing the change that we're seeing. It makes it easier for us as the scientists and experimenters to pinpoint, in this case, the pollution and say, that's it. That is what is causing these salamanders to grow up with the weird tails. So that's why it's so important to have just the one changing variable in our experiment, so that if we find different results in the two different settings, we know that it has to be due to changing that one thing. That's the only thing that was different, so that must be the reason behind the change. All right, so obviously we want to have a lot of control over our variables. If we only want this one thing to change and other things to not change, then we have the thing at the end that we're measuring. We have to make sure to account for all of those and be ready to take care of all of those when we go into our experiment. So there are kind of three major groups of variables that we deal with as scientists. So we are changing that one thing, like I said, and then measuring or observing what happens, right? We want to have some sort of results to go off of this experiment. So the thing that we are changing, the thing that we as the experimenters, the scientists, are going in and messing with, is called the independent variable. So the independent variable, in the case of our salamanders, it would be the pollution that we added, right? Everything else was kept the same, but as scientists, we went in and changed the amount of pollution that's in there. So that is our independent variable. So this is the one thing that we're changing in the experiment. It's called independent because it doesn't depend on any other variables that are part of the experiment. It only matters what we're setting up at the beginning. So the thing that we're changing, the thing that we're adjusting or altering is the independent variable. If you did that experiment with the plant fertilizer and you wanted to see if adding fertilizer to plants helped them grow better, then the independent variable would be adding the fertilizer or the amount of fertilizer that you added. It's the thing that we're changing at the beginning of the experiment. The experiment goes through, hopefully we had some different results, different measurements come out from our changes. And those observations or measurements that we make in the experiment are called the dependent variables. So the thing that we're measuring or observing at the end of the experiment is the dependent variable. So in our salamander example, the dependent variable would be the number of salamanders that grew up with crooked tails. That's the thing that we're observing at the end of the experiment. In our plant fertilizer example, the dependent variable would be how large the plants grew when the fertilizer was added. So like the height of the plant or how many flowers that it produced at the end of the experiment, that would be our dependent variable. So it's called dependent because it depends on what we do with the independent variable, right? Whether or not you're a salamander that has a crooked tail depends on if you were given the pollution or not. How tall that the plant grows depends on if it was given fertilizer or not. So that's where the names come from, but the main thing to remember is the thing that we are changing, the thing that we're altering, messing with, is the independent variable. The thing that we're measuring or timing or counting at the end of the experiment, that is our dependent variable. So independent, we are altering it. Dependent, we are measuring it. So there's one final kind of larger set of variables that we have to deal with, and those are called control variables. So the control variables are the things that we are keeping the same across all the different parts of our experiment so that they don't have any sort of effect on the outcome that we don't want them to have, right? So in our salamander experiment, this would be things like when we went back and made sure that the temperature was the same and the humidity was the same and the water was the same, and we put the same amount of salamanders into each chamber, and we make sure that they're all the same age at the same time. Those things are control variables. Things that we're trying to keep the same between all of our experiments so that only the independent variable is having an effect. We don't want there to be any sort of effect from the temperature or the humidity or anything like that. So in like the plant fertilizer example, control variables would be things like the amount of sunlight that the plants get each day should be the same for all the groups. The type of soil that they're potted in, how large of a pot that they're put into. It should be the same plant that's getting the no fertilizer versus the fertilizer groups. All those things should be kept the same so that only the independent variable is affecting the outcome. The control variables you want to keep the same across everything because it makes it easier to do that comparison, right? Like we were saying earlier, if we change lots of things in the experiment at the start, then it's hard to compare between the groups because we don't know what's causing the changes between the groups. So if we can keep all that stuff the same and only change that one independent variable, then it becomes a lot easier to compare the groups and say, this one without the fertilizer only grew 6 inches. This one with the fertilizer grew 14 inches. The temperature was the same. The sunlight was the same. The plant and the soil was the same. It must have been the fertilizer that caused the taller plant to grow taller. So by keeping all of those controls all the same, it makes it easy to compare between the different experiments, the different groups that we have as part of our experiment. So we have our independent variable, the thing that we are changing or altering as part of the experiment. We have our dependent variable, the thing that we're measuring and observing and collecting data on in the experiment. And we have all of our control variables, things that we are trying to keep the same between all of the different parts of the experiment. So those are kind of the 3 major categories of variables that scientists have to keep track of when they're setting up or when they're designing an experiment. So I'm going to have you guys help me design a really simple experiment here. I'm going to give you kind of the basic premise of the experiment, and then I want you guys to help me figure out what are our independent, dependent, and control variables. So the experiment is going to go something like this. You want to figure out the fastest route to get from school. So we're pretending like everybody's here at school in person on a daily basis, like back before COVID times. Everybody's at school, and you want to know what is the best way to get to your house from school after class is over. This example says walk, but I know for us here, it's mostly driving, right? Most people drive from the school to their house. And you want to figure out the fastest way that you can take home from school. Usually you have like the same route that you take every day. You hop on the highway, you go to this highway, then you get off at this exit and go home. But what if there might be a faster way, a better way to get home? So when you come to school in the next week, Monday, Tuesday, Wednesday, Thursday, each day you're going to take a different way home. And you're going to time yourself to see how long it takes you to get home. So on Monday, maybe instead of turning right, you'll turn left out of the school and go to this road and then straight down that road and then make a left on that road. And then on Tuesday, you turn right at first and then turn left and then go straight for a while. And then on Wednesday and Thursday, you repeat with different routes. So each day you take a different route home and time how long that it takes you. So as soon as you walk out, as soon as you get into the car, you start the stopwatch on your phone. And as soon as you pull in to your driveway or parking space at your apartment and get out of the car, you stop the stopwatch. So you're going to compare how long does it take you to get home when you go route A on Monday versus B on Tuesday versus C on Wednesday and D on Thursday. So you want to make sure that it's the one that works best and fastest for you. So you're not going to have your friends drive the route with you and compare to all of their times. You're going to be the only person who is driving. You're not going to be using a different car each time, right? Because cars sometimes are different in terms of how they speed up and slow down and stop and stuff like that. So it's not like you're going to be borrowing your brother's sports car on Tuesday and your uncle's motorcycle on Wednesday and your mom's minivan on Thursday. You're going to be using the same car each day. And obviously, I guess different people end school on different times, but you probably end school at the same time on each day. So you'll be leaving from the Excel Center at the same time, doing it yourself, getting in your own car, changing up how you get home, and then timing how long that it takes. All so that you can figure out what is the fastest way to get from school to your house. So I want you guys to help me figure out what are our different sets of variables in this experiment. So up first, we have our independent variable. So what is the independent variable in our drive home experiment?
Maybe like the car.
The different routes that you'll be taking.
Hey, Ashley, you nailed it. So yeah, the independent variable is the different ways that you're getting home, because that's what you are changing as the experimenter, right? You are changing how the exact way you take to get from school to your house. So the one thing that the scientist is changing in the experiment is the independent variable. Good one, Ashley. All right, up next we have our dependent variable. So what is the dependent variable in our driving home experiment?
The time it takes you to get to point A to point B.
Exactly. So remember, the independent variable is what we're measuring or what we're observing in our experiment. So in this case, we're measuring how long it takes to get from school to house. That's the result of our experiment. So yeah, exactly. Our dependent variable is how long does it take you to get from school to your house? That is the thing that you are measuring as part of your experiment. All right, and now somebody who is not named Ashley, what are some of our control variables in the experiment? What are things that we're keeping the same across the whole experiment?
Trying to get to the same place. Each way is to the same road, I mean, to the same destination.
Yep, so Kevin points out we're starting and ending at the same place each time. That's staying constant across everything. So that's a good control variable. What else is staying the same each time? Nasima, I think you mentioned one earlier.
Like your car?
Exactly. You're using the same car each time, right? It's not your friend's car. It's not your mom's minivan. It's not your uncle's motorcycle. It's your car. The same car is being used each time that you drive home. Good. Excellent. Other control variables would be like it's you are the person that is driving, right? It's not like on Monday you tell your friend like, okay, meet me at my house and time yourself how long it takes to get there. Because maybe your friend drives differently than you do. So you are the same person doing this experiment, doing the driving each time. Awesome. So yeah, changing up how we get home is the independent variable because this is the thing that we as the experimenter are changing as part of the experiment. How long it takes to get home is the dependent variable in our experiment. This is the thing that we are measuring. And then finally, again, this says the same walker, like you're walking home, but the same driver, same car, leaving at the same time of day, starting and ending at the same spots. These are all examples of control variables. So usually each experiment has a lot of control variables that scientists have to keep track of in order to try and make sure that they all stay the same across all the different experiments that you do. All right, very, very nice. Good job, everybody. Thank you for your help. Does anybody have any further questions about independent, dependent, or control variables? Anything that you're still a little confused about or want some more explanation on?
Yes, Mr. Michael, please, the variables one, the dependent variable.
So our independent variable is the thing that the scientist is changing at the start of the experiment. So with our salamander experiment, we're changing if there's pollution in the environment or not. If we're doing an experiment with plant fertilizer, we're changing if the plant gets fertilizer or if it doesn't. In our driving home experiment, each day at the start of the experiment, we're changing how we get home, right? So the thing that the scientist is changing at the beginning of the experiment is the independent variable. So this we are changing on purpose. We're purposely altering part of the experiment to make it change. The thing that we are measuring in the experiment, the data that we're collecting, what we're observing in the experiment is the dependent variable. So in our salamander example, our dependent variable is the number of salamanders at the end of the experiment that have curvy, crooked tails. That's what we're counting at the end of our experiment. In the fertilizer experiment, our dependent variable is how tall or how wide our plants grew at the end. It's what we're measuring after we add the fertilizer to the plants and let them grow for a couple weeks. We then measure how big did they grow. And that size is the dependent variable. It's what we're measuring as part of the experiment.
So like basically the result that we're looking for?
Exactly. The dependent variable are your results.
Okay, thank you.
No problem. So yeah, usually the independent variable is something that the scientist sets up at the beginning of the experiment. The dependent variable is what the scientist measures at the end of the experiment. All right, great question. So speaking of dependent variables and measuring things, all of that information makes up our data. So data is essentially the results of the experiment, what you measured or counted or observed as the experiment went on and reached its conclusion. So there are kind of two different categories that your data can fall into. You could have probably the most common type of data is called quantitative, which just means that it's based on numbers. How many salamanders had the crooked tails? Exactly how tall did the plants grow? How many seconds did it take to get from the school to home? These are all quantitative points of data. They're based on numbers, measurements, stuff like that. Qualitative is kind of more descriptive type of data. So if you're doing an experiment and you saw something change color as part of it, it went from red to blue. That's an observation. It doesn't really have numbers associated with it, so it's qualitative data. If you're eating food over time and it becomes more like gross and more rotten and stuff, and so it tastes worse and worse, taste you can't really put into numbers. So that would also be kind of a qualitative data point. For the most part, scientists aren't huge fans of qualitative data. We really like quantitative, hard numbers, because numbers, it's easy to compare one to the other and get your answer. But regardless of what type of data you have, those are what make up the results of your experiment. You did the experiment, You change the independent variable, let it go for a while, and now you're measuring what your results are. You're getting that dependent variable. So we use our data in order to form what's called a conclusion. So in a conclusion, we use the data, we use the results that we got from the experiment in order to answer our hypothesis. Say either our hypothesis was right, it is the pollution that's causing the crooked tails, or our hypothesis was not supported. We put in pollution, but we didn't see any change in the tails. So a conclusion is what answers our original kind of question, our original hypothesis. So a good conclusion is usually made up of two parts. It has your final results, and it talks about whether or not your hypothesis was supported. So we found that we had more crooked-tailed salamanders in the pollution chamber versus the clean chamber. This supports our hypothesis that pollution causes salamanders to grow with crooked tails. I found that it took me the exact same amount of time to get home, no matter which route I took from the school to my house. So my hypothesis that changing up the route would get me home faster was not supported. I didn't find any evidence that going home a different way made it faster to get home. I gave some plants fertilizer, and the plants with fertilizer grew more flowers than the plants that did not have fertilizer. So my hypothesis that adding fertilizer will help make flowers was supported. No worries, Nevin. It happens all the time. Don't worry about it. As long as you can get back in, that's the important thing. So that is what the conclusion, the final part of the scientific method is here for. To say, essentially, what did you find? And did you support your hypothesis? Was your guess correct? Or was your guess a little bit off, not quite right? You found some evidence that did not support what you thought was going on. That is what our conclusion is for. And then finally, the last thing that I want to mention is that usually in science, we can't just do an experiment one time and say, oh, this is the result that we found. Our hypothesis was supported. Good job, everybody. We can go home now. Instead, we want to make sure that there are lots and lots and lots of data points, lots and lots of experiments that we did to show we got the same answer every time. So that's where retesting experiments comes in. It helps us to verify the results and take kind of an average result instead of relying on just one. Like, for example, if we were doing the driving home experiment, and on that particular day that you were driving home, there was a really bad accident on I-35, and it took you a lot longer to get home than normal. That doesn't mean that that's the worst way to get home. It just means that on that one day, there were some things that you couldn't control that may have affected your results. So there's always going to be things outside of a scientist's control that can affect results. But by doing lots and lots and lots of tests, doing the same experiment 10, 15, 20, 100, 1,000, 10,000 times, you can get really, really strong data about whether or not your hypothesis was supported. So for a really, really great modern example of this, we can think about the COVID-19 vaccines from Moderna and Pfizer and Johnson & Johnson. In order to get the okay from the government, the Food and Drug Administration, to be released as a vaccine, a company has to go through what's called a phase 3 clinical trial. And this is where they test their vaccine on tens of thousands of people because they're looking for two things. One, does this vaccine help stop people from getting COVID? And two, are there any really bad side effects that this vaccine will cause in people? Because if it's great at preventing you from getting COVID, but everybody who gets the vaccine loses their right arm, that's probably not a super safe vaccine that people are going to want to take. So they have to do it on thousands and thousands and thousands of people. Because imagine that a new vaccine came out and they said, we're really excited about this vaccine. It is super duper effective and super duper safe. We tested it on 2 people in San Antonio and both of them were fine after they got the vaccine. So we think that everybody will be fine. You'd probably be a little bit nervous about getting that vaccine, right? That's because maybe there's rare side effects that only some people are going to have. Or maybe those people were already really immune to COVID and the vaccine actually didn't help them all that much. So instead, these companies have to do these tests on thousands and thousands of people in order to show, hey, we gave 13,000 people our COVID vaccine and only 4 of them ended up getting COVID, which is way less than the 400 that got COVID in the non-vaccine group. So the more data points, the more retesting you can do of your experiment, the stronger your results are at the end. The more you can say, hey, this definitely supports my hypothesis that this is a safe and effective vaccine. So by having lots and lots of retesting for vaccines or for whatever type of experiment that you're doing, you can reduce some of like the random chance that was always there that could mess with your experiment and give you a really strong average amount of results. So if we put this, now that we've talked about all our different steps here, if we put it all back into the kind of general scientific method, we have some sort of problem that a scientist is interested in. You're really interested in salamanders. You go out and you observe that salamanders in certain areas have these weird tails. You make some observations about this thing that you're interested in. You then use those observations to form a hypothesis. I think that if salamanders grow up in a polluted area, then they're gonna develop these weird crooked tails. You then can design a good testable experiment in order to test out your hypothesis, right? You want to know, you want to get the answer to your guess, to your question. So you make sure that you have that one single independent variable that you're changing. You figure out what you're going to measure as part of your dependent variable, and you do your very best to make sure that all of the control variables are accounted for. There's nothing that's changing between your different experiment groups besides that one single independent variable that you're interested in, whether that's pollution or fertilizer in plants or the amount of time it takes to get home or the way that you get home. So you run your experiment, you take lots of observations and collect a lot of data through it. You measure your plants, you count up the crooked-tailed salamanders, you time how long it takes to get home. And then you use that data, you use that information to form a conclusion. Usually referencing back to something about your hypothesis. Either my hypothesis was correct, the salamanders in the pollution grew up with much more crooked tails than the normal salamanders, or my hypothesis was not supported. I gave a whole bunch of fertilizer to these plants and they grew just as well as the plants with no fertilizer. My hypothesis was wrong. I'm going to have to come up with a different experiment to try and help answer some different questions. And then hopefully you did lots and lots, multiple versions of your experiment in order to try and get kind of the most robust and the strongest data possible. So this is the same basic set of kind of guidelines that scientists use in order to come up with an idea for their experiments. Figure out some sort of question they want to answer, a problem you want to solve. Take a guess on what you think could be the answer. Run some experiments and gather data and use that to see if your guess was correct. All right. Does anybody have any questions about any of these parts of the scientific method? The hypothesis, the variables in an experiment, making a conclusion from your data, anything that anybody still wants some explanation about?
No, thank you, Mr. Michael.
Alrighty. Well, I think that brings us to the end of our notes for biology class today. Again, please remember, if you're able to do it, to finish up that writing assignment for today, take a picture of it and text it, email it to me, or post it on the Google Classroom. And then don't forget, on Monday of next week, before we meet again, your syllabus quiz will be posted. Super simple 5-question quiz about like the basic information from this class. So make sure you're keeping an eye out for that on Monday. And then on Tuesday, we'll really start to get into our first real set of biology notes.